So now we will learn about the mechanism of T-cells thymic selection in detail. We know that the T-cells in the circulation must have proper compatibility with the MHC antigen complex. Only then it can recognize the antigen and perform proper action against the invading pathogen. Also, in our previous videos, we have mentioned that T-cells are highly MHC restricted, and so this restriction is determined even at the time of T-cell development. So, by this selection, the thymus ensures that most T-cells that are released into the circulation function in conjunction with the corresponding major histocompatible complex genes of the body's immune system, and identify only self-MHC with foreign antigen. But do not identify any self-antigen as foreign material, so to avoid autoimmune reactions. After double positive immature T-cells formation in the cortex of the thymus, they now undergo the process of selection within thymic medulla. This selection of T lymphocytes can be positive or negative. When we say positive selection, that means that lineage will continue to be mature T cells. And if they are negative selected, that means those cells are eliminated or cell lysis may occur. So, what happens is that, during their selection, immature T cells encounter various populations of cells that express MHC and self-peptide. Immature T cells first encounter cortical thymic epithelia, which are involved in positive selection. So what is this positive selection? Well, to continue along the developmental process, the DP cells must first make a successful interaction with MHC molecules on thymic cortical epithelial cells, or those epithelial cells that are found in the cortex of the thymus. But most DP cells die in the cortex through apoptosis because they lack TCRs capable of interacting appropriately with self-MHC. So, these cells will undergo apoptosis. Only DP cells that have TCRs that recognize self-MHC with medium affinity will survive and thus are said to be positively selected. So, this is positive selection. Then what is negative selection? The pool of cells generated by positive selection in the thymus is self-MHC restricted, that make appropriate strength bond with self-MHC and foreign antigen. But it also includes some self-reactive cells, which could recognize self-peptide plus self-MHC. These self-reactive cells must be eliminated or the host risks autoimmune disease. Elimination of self-reactive cells involves a process called negative selection. So, which type of cells will be positively selected or which ones are negatively selected, let's understand via these figures. Here in all these figures, the newly developed TCRs are interacting with the MHC molecules on the epithelial cells. Now, one of the following events may occur. The thymocytes may be unable to bind with the MHC molecules via the T-cell receptors, because of mismatched binding. Like in case 1. And we know that for the prevention of an infection it is necessary that TCR must bind with the MHC molecules, but it is not the case in first figure. So, these T cells are not properly programmed, and thus they need to be eliminated. And this elimination is not done by actively killing the cells, but by an endogenous suicide program that is referred to as apoptosis or programmed cell death. Then in the second case, we can see that the TCRs are now properly binding with the MHC molecule. But here this binding is too strong. And, if the bond between the T-cell receptor and the MHC molecule is too strong, a cytotoxic response to the body's own antigen-presenting cells may later occur. So, even in this case, T cells will not survive and they will be destroyed via apoptosis. Now, in some cases, like in this third figure, the T-cell receptors and the MHC antigen may match, but the receptor recognizes an endogenous antigen instead of an exogenous antigen. And so, it may result in autoimmune reactions, if these cells mature. These cells come in encounter with the dendritic cells that possess most, if not all, surface antigens known to exist. So, the T cells that react with one of these autoantigens will be negatively selected and thus undergo apoptosis. Then comes the fourth figure: only those cells that recognize the matching MHC molecule with exogenous antigen and form a moderately strong bond with it will be allowed to fully mature and pass as fully functioning T cells into the circulation. So, that's how p-thymocytes get selected. But now there is another type of selection required for further T cells development, where the selected double positive or DP cells are transformed into single positive or SP cells. That means, at this stage, now the thymocytes have to decide their particular cell lineage, that can be either CD4+ or CD8+. So, during this selection, it has to be decided whether the DP cell recognizes the antigen presented on MHC class I or class II molecules. If you recall our video on MHC and antigen presentation, you will remember that the CD8 molecule recognizes MHC class I and CD4 recognizes MHC class II. So, the DP cells that recognizes antigen on MHC class I stop or reduce expressing CD4 and become single positive, SP, CD8+ cells. In contrast, DP cells that recognize cell self-antigen on MHC class II stop expressing CD8 and become SP CD4+ cells. Within the SP, CD4+ cells, the 10% of cells with the highest affinity for MHC class II antigen activator transcription factor called FOXP3, which commits these cells to a different differentiation pathway, and they eventually mature as natural T regulatory cells. Whereas the rest of CD4+ cells then forms the helper T cells. And SP CD8+ cells makes the cytotoxic T cells. So, this was the detailed mechanism of T cells development and selection. You can get a full picture from this slide. It has been estimated that 2/3 of the cells that survive positive selection in the cortex are subsequently deleted by negative selection in the medulla. Consequently, a very small number of the original thymocytes emerge into the periphery as mature T cells. So, this was all about the development and selection of T cells. I hope you would have learned a lot from this video. And if you want to watch more such interesting and full of knowledge videos, then keep visiting sqadia.com.
Discover a wealth of accurate, up-to-date medical videos for in-depth learning, anytime, anywhere. Elevate your medical expertise with sqadia.com.